Okay, so I'll continue on, lecture 15, the third part, I guess. From now on, I'm gonna make a very short videos, chop into a much smaller segment to help you understand better. So for this segment, what I'm gonna do is to tell you about the mesotherm induction and then mesotherm patterning. I think that's I'm going to focus more on. Uh, you know, what I'm going to focus more on is okay how the endoderm is specified. Okay, okay, how the endoderm is specified first, and then we'll talk about this Peter Newcook. Okay, I think I might have misspelled his name. Okay, so to demonstrate the mesoderm came from endoderm, ectoderm, and then endoderm is one of the signaling center. Okay, so so which means that we have the let's go back to very, very early stages, and then we only have a zygote, yeah, one cell, and then they divide, and then which means that they had a two-layer first. So then how are we gonna define this two? Okay, so looks like we are kind of like going back to future first. Okay, so it is the mesoderm is coming from ectoderm, and then ectoderm is receiving the signal and then turning into the mesoderm. So that is the principle. Then we have an ectoderm and then endoderm here. Then yeah, this is endoderm and this is ectoderm. And which layer has to be specified first? Okay, and then we really need to define it. And that is, if you think about it, in mesoderm induction purpose, then most likely you need to define this because endoderm is the one that giving the signal. So there's a signaling center. So you need to define those first. So scientists really work very hard. What is determining? Not only those three germ layers later, okay, but the what is determining this and this? That is the one of the big question. And then I think that's veggie T because this is the coming from vegetal hemisphere, right? So veggie T, they found the transcription factor, and then that is really important in determination of this endoderm area. So veggie T is a transcription factor, and then whose mRNA is localized to vegetal code, just like a veggie one, we saw it. And then this veggie T protein is produced in vegetal blastomeres to the fertilization, and then it is required for induction, mesoderm induction, because you have to have endoderm, isn't it? I mean, ectoderm doesn't really matter, but even if you have ectoderm, if you don't have the endoderm, then you cannot make the mesoderm. Okay. So the veggie T is really required for endoderm specification. So what you can do is that also you can do the removal assay of veggie T. And then so you use this antisense veggie T oligo, you inject them and then you wipe them, you wipe out veggie T mRNA. Then what will happen is that you can see in here, okay, this is the veggie T, anti-sense veggie T mRNA, and then when you inject them, and then you can see SOX17, HOX8, and then insulin. These are the mesoderm endoderm marker, and then those are disappeared. I think it has endoderm markers from here to here. So then what are the other lane? I think that this is the loading control kind of thing. And then control RT is that without the PCR. Okay, so this is not something fake. So that's why you are showing here. And then control is that the without RT. And then they are able to turning it into endoderm. So it is showing endodermal marker. Okay. And then oligo is the real antisense VEGF when you inject them, and then you are able to make them disappear. Of this mesoderm marker, you see no bands in these three things. So no endoderm. And then this is the you add one more oligo here. Okay, so that is a VEGF oligo. So what it is that you add the inhibitor of inhibitor. Okay, so VEGF is like a sense RNA. Okay, so the sense RNA will bind to the antisense. Then you are basically, it will wipe out the inhibitor that the causes this make the endoderm marker disappear. And what you found was that okay, when you add the inhibitor of the inhibitor, the marker came back. So trying to show you that is the specific band. Okay, so anyway, VEGF specify the endoderm. So next is that okay, we need to demonstrate the inducing mesoderm is by the endoderm, and then by changing the part of ectoderm into mesoderm. Okay, so Peter Newcomb. Okay, I read his obituary about like several years ago. So he passed away. He's he's from Denmark. So Newcomb actually did like a carving out, using razor blade. You cut out this part and then culture them separately, and then which part is developing into. Because this independent event, cell autonomous, it's a kind of like a hard concept. We'll talk about it a little bit later. But anyway, he did separate them into culture dish, and then they found that okay, here is the mesoderm, and then depending on the side, they can have a different type of mesoderm coming up. And animal cap is the ectoderm. Animal cap is the ectoderm, so you have the ectodermal tissue show up, and here's the endoderm, so endodermal tissue showing up. So the initial experiment is this. So he did the animal cap assay. That's what I say. And then you have animal cap, very, very top of this, ectoderm. You place them in there, and then you have this chunk of endoderm you carve out, and then you place it, and you put it together. You co-culture. Okay, right. And you find that okay, inside the mesoderm is developing, and you need to have some markers so that okay, mesoderms have the same marker that ectodermal cell carries. So that's why mesoderm is from actually ectoderm. Okay, right. Okay, so that was his demonstration. Okay, so once again, this vegetal cell is signal, isn't it? And then maybe changing that. So he did this experiment. He put the some kind of barrier between this ectodermal cell and then endodermal cell, but they are porous. I mean, you think of like a saran wrap or something. Okay, a saran wrap actually has a small holes in it. Okay, so those porous membrane you put it there, they were still able to form this mesoderm. So which means that substance that's coming from endoderm is something that's secreted. Okay, secreted molecule. So that's why they can pass through. And then they found he tried to do it's kind of weird experiment, but how about what he did is that he grinded up this animal cap, and then he remove all this cell-to-cell interaction, and then he put very little number of cell on the top of this endoderm, and then in that case, they don't change into the mesoderm. So he controlled the number of cell, and then what he found is that, okay, at least you need to have more than 200 cells coming over to the mesoderm, endoderm, and then they are able to change to a mesoderm. So that is the community effect. So I want you to, think about this. So I'll give you 10 seconds to think about it, okay? Okay, 10 seconds. Okay. So, which means it's the community effect. Okay. So what it means is that looks like whatever inducer is also produced from ectoderm. So mesoderm inducer is also produced from ectoderm itself. So maybe initial signal comes in this case. Okay, and then initial signal from endoderm comes into this cell, okay? And then they are changing into mesoderm, but also they are producing mesoderm inducer by themselves. And then that kind of, signal has to be there. So that has to reach a certain level, above the threshold level. But if cells numbers are less than 200, then probably they may not reach that level, okay? I mean, that is my, interpretation. I think that you might want to read, this chapter about community effect of the nuclear experiment in the Warburg textbook. But that is my, interpretation. If you have problem, let me know. Okay. So and next is that they check the time. So you have the they took out this animal cap in a different period of time, 1 hour, 2 hours, 3 hours. Too small is not gonna work. And then 4 hours after fertilization, then it starts to change to mesoderm. But if you isolate this mesoderm, ectoderm chunk, which is an animal cap, after 11 hours of the fertilization, then mesoderm induction doesn't work. And then they just take on the ectodermal phase. So probably after this, they're going to be locked in to the ectoderm. So they cannot be changed. So probably this is the specification process going on in here. Okay, right. Okay, and then also what they found is that, so that's about 7-hour period of time. And what will happen is that, you need to induce them at least 2 hours. So you need to put them together at least 2 hours to make the thing happen. So, it's perfect. It makes perfect sense. It takes some time, isn't it? Right. Okay. So from this, and then fate map and everything, and then what they found is that, default map is right here. Okay. So actually, you isolate them before the induction or something, but they produce this induction. Okay, they produce some kind of basic mechanism, produces mesoderm, and then produces also dorsal mesoderm. But if you do the fate map, so you just let those cells in there, and then if you don't isolate it, some kind of signaling event after the mesoderm induction, then they will have this patterning on top of this induction, but they also have this pattern. So dorsal to ventral pattern shows up. So this patterning is also what's make this kind of like a process really special. That I'm going to finish, here, and then I'm gonna continue next. Today I talked about endoderm induction, vegetative function, right? And then, mesoderm is produced from ectoderm by the endoderm. And then I talked about a series of this Peter-Newcomb experiment. I'll just stop here and I will see you in next segment.